Hi, this is Abem, and I'm going to be showing you the transduction pathway of a platelet-derived growth factor, also known as PDGF. Here we have a plasma membrane with two monomers of PDGFR, a type of receptor tyrosine kinase monomer. These contain tyrosine kinase domains that you can see with the bumps alongside the monomers. We can see a homodimer, a protein composed of two identical polypeptide chains of PDGF, come in to bind both monomers of PDGF. This leads to the receptor undergoing dimerization, or the process of bonding. Now, the molecules will lead to the cross-postulation of the PDGF kinase receptors. This process of these monomers leads to the receptors activating. Due to this, countless signaling pathways are created inside of the cell. This ends the process of normal PDGF activation. Now we are on to a new scenario known as chromosomal translocation. This occurs when a chromosome shatters and rearranges itself incorrectly. This can be a cause for Down syndrome. In our case, this leads to a mutation in our PDGF receptors. One of the big differences in this mutation is that part of the PDGF receptor is replaced by our TEL-ETV6 proteins. This produces a slightly different receptor for PDGF, and the TEL-ETV6 dimerizes the two monomers by itself. Due to this change, signaling starts without any outside ligands. All of this leads to the activation of the tyrosine kinase receptors, but since the signal cannot be turned off, this leads to cancer, which ends our modified PDGF process. Thank you for listening to our presentation.